For this video, I'm going to be talking about antibody-mediated effector functions. And to start out, I'm just going to talk a little bit about antibodies. So antibodies are also known as immunoglobulin, which is Ig. It's a large Y-shaped protein produced mainly by plasma cells that is used by our immune system to neutralize pathogens such as bacteria and viruses. And they are secreted by B cells of the adaptive immune system, mostly by differentiated B cells called plasma cells. And then this is actually just a picture of what an antibody is, and as you can see, it's the Y-shaped. This is the antibody, and then this is the antigen binding site, and then this is the antigen. And as you can see, it's just giving you an example of an antigen binding to the antibody. And this antigen is the same shape as the antigen binding site. And in order for these antigens to fit into this antibody, it would have to have the same base shape. And so, like, if this antigen wanted to fit into this antibody, the base of this antigen would have to be the same as the antigen binding site. So none of these antigens would be able to bind right here because they wouldn't fit and nothing would happen. So activation of effector cells. To combat pathogens that replicate outside cells, antibodies bind to pathogens to link them together, causing them to agglutinate, which means clump together. Basically, when I hear that word, I think glue together because it kind of sounds like a glutinate. It kind of sounds like glue. An antibody has at least two paratopes. It can bind more than one antigen by binding identical epitopes carried on the surface of these antigens. And by coding the pathogen, antibodies stimulate effector functions against the pathogens or against the pathogen in cells that recognize their Fc region. And I'm going to talk a little bit about what Fc receptors are in a little bit. Those cells that recognize coded pathogens have Fc receptors, which, as the name will suggest, they interact with the Fc region of IgA, IgG, and IgE antibodies, and so all of those are antibodies: IgA, IgG, and IgE. The engagement of a particular antibody with the Fc receptor on a particular cell triggers an effector function of that cell, and some of those include phagocytes with phagocytosis. Mast cells and neutrophils will degranulate, and then natural killer cells will release cytokines and cytotoxic molecules. That will ultimately result in destruction of the invading microbes. So the ones that I just mentioned will result in the destruction of the invading microbes. The activation of natural killer cells by antibodies initiates a cytotoxic mechanism known as antibody-dependent cell-mediated cytotoxicity, or ADCC. That's a lot easier to say. This process may explain the efficiency of monoclonal antibodies used in biological therapies against cancer. So basically, biological therapies are like the things that we're trying to come up to help slow down cancer, cure cancer, things like that. The Fc receptor are isotype specific, which gives greater flexibility to the immune system, invoking only the appropriate immune mechanism for distinct pathogens. So only the appropriate ones, or the ones that will work. Antibodies act by a number of mechanisms, most of which engage other. Sorry, I don't know why it does that every time. Antibodies act by a number of mechanisms, most of which engage other arms of the immune system. So they basically trigger other parts of the immune system. They can simply block interactions of molecules, or they can activate the classical complement pathway, and that's another video that I talked about, the classical complement pathway, known as complement-dependent cytotoxicity, or CDC, by interactions of C1q on the C1 complex with clustered antibodies. And when it's like clustered antibodies, it's just a bunch of them together. And antibodies also act as a link between the antibody-mediated and cell-mediated immune responses through engagement of Fc receptors. So Fc receptors are key immune regulatory receptors, connecting the antibody-mediated humoral immune system, humoral immune response to cellular effector functions. So basically, the antibodies in our body, like I said, they are secreted by B cells of the adaptive immune system, mostly by the differentiated B cells called plasma cells. So there are antibodies in the innate immune system, but the adaptive immune system secrete the B cells, or B cells are secreted in the adaptive immune system. And I don't know if you caught the effector function of the cell, but I'm going to repeat those again. Phagocytes with phagocytose, mast cells and neutrophils will degranulate, and then natural killer cells, or NK cells, is what they're mostly, like you can find them that just say NK, but those are natural killer cells, will release cytokines, or cytokines, and cytotoxic molecules, that will result in the destruction of the invading microbes. So anything that's invading you, those will stop it and basically break it down and just quit altogether. And then the Y-shaped, again, those are just antibodies, and they're just going to be Y-shaped. Like all antibodies are Y-shaped, and then the antigen binding site, you have to have an antigen that matches the binding site.